mutations, and we started last time looking at mutations that can be induced, that is, what kind of mutagens can lead to increased mutation rates, primarily because of affecting the DNA in some way or another. So we were talking the last time about alkylating agents. These are one of the categories of DNA reactive mutagens that react at some point, to make a change in the DNA. They react at some point with DNA. They don't have to be interacting with replicating DNA, but obviously to fix any kind of DNA damage or mutational change, it would require DNA replication to make that a full mutation. You see it through the process of DNA replication. So we're looking at this example of the alkylating agents that add alkyl groups to areas on some of the bases. And the net result of this is that it alters those modified, those alkylated nucleotides undergo a tautomeric shift more often than a non-alkylated, non-modified base. And so it increases the likelihood that there will be a transition mutation as a result of that tautomeric shift. Same mechanism that we've seen before with just the transition as the normal isomer change in the DNA normal bases, but here that change happens with a slightly higher frequency, so it increases that probability. It also can result in DNA damage because sometimes that alkylation on the base makes that glycosidic linkage unstable. That can result in loss of the base, right? So if it's a purine, a guanine is a prime target for alkylation, it can result in depurination, and that depurination can result in the production of transversions. Remember, that was the primary origin of transversions that we saw. There can also be damage to the DNA backbone. It can cause breakage in that single strand. It's probably not as common as the mispairing or depurination. Okay. And as I said before, these alkylating agents, it's a very significant group of chemical compounds that can impact the DNA in this way, and they can have pretty broad impacts. Okay. So another group in this category of DNA reactive agents are the intercalating agents. So intercalate, to fit in between. And that's what these agents do. Their size, they're flat and they're hydrophobic, and their size is pretty similar to the size of a normal nucleotide base. And so as a consequence, they're able to squeeze their way in between the stacked bases in the double helix. Good example are acridines, and you see some of these compounds down here. You see the ring structure that is similar to some of the base structure that you are familiar with. Because they're able to stack in between those bases, they don't have to have the DNA replicating at the time, but if they're in that DNA at the time that the DNA starts to replicate, it can result in bulging or looping of, you notice it here in stacked in between the bases of the double helix, it can result in bulging or looping or that kind of sliding of one strand relative to the other that we talked about with repeated nucleotides that was the origin of insertions and deletions. And so typically these intercalating agents cause or are associated most commonly with insertions and deletions because of that strand slippage or misalignment during DNA replication between the template strand and the daughter strand as a result of that integrated agent, that compound there. Many of you have seen these agents before. So how many of you have run DNA electrophoresis gels where you stain them to get this really nice fluorescent color of the band? That staining is accomplished by an intercalating agent called ethidium bromide. It's the most commonly used DNA dye for molecular biology. And you see this nice fluorescent hat. Ethidium bromide is an intercalating agent. It's a very potent mutagen, which is why you wear protective clothing, goggles, and you dispose of it appropriately. It's also a strong carcinogen, consistent with that generalization I made at the beginning that most mutagens are also carcinogens. So this is one of the effects. We also have similar agents that are used to stain tissues. in whole organisms, particularly useful in embryos. So this is DAPI, which is a fluorescent intercalating agent that is used all the time in developmental biology. Whenever you're doing tissue staining, it stains, it intercalates in the DNA, which means that it's going to show a nice fluorescent stain in the nuclei. So these are embryos from a variety of different model organisms. This is a Drosophila early embryo. You can see these round stained blue dots. Those are the nuclei in this embryo. At this stage in early development in Drosophila, the whole embryo is a syncytium, meaning it's one gigantic cell. There are no cell membranes that separate these nuclei. They're nuclear membranes, but during this stage, the nuclei are dividing even outside of kinesis. That happens later when the membranes form between the cells. So it looks like this gigantic cell with all these different nuclei. This is another embryo of a model organism. Anybody know what this is? Anybody work with it? It's C. elegans, Cenorhabditis elegans. It's this little soil nematode. How about this one? Anybody recognize that? Probably not so common. Still a valuable model organism, especially for neural development in the early days. That's a grasshopper embryo. Yeah, it sort of looks like a grasshopper embryo. All right. So you see these kinds of agents that result in damage to the DNA, mismatching of the DNA, so tautomeric shifts that result in transitions, damage that can result in transversions, damage that can result in structural modifications of the DNA. And so it should occur to you that we really ought to be able to recognize and identify when we encounter such potential and powerful mutagens in our environment. And that's a major occupation every time some new chemical is developed, either in as a means of food preservation or in food production or in the environment in terms of pesticides or any other kind of agent that we expose our environment to, expose ourselves to. So a major issue becomes how do we detect these mutagens? How do we recognize these potential mutagens in order to then limit our exposure to them, limit the interaction with them? We all know that irradiation causes problems, it causes damage, and so it's a very widespread practice now to use sunscreen so that you're protecting your skin cells from the damaging radiation of sunlight. But what about some of these chemical mutagens that we're exposed to all the time? So there are a number of different ways that have been developed over the decades to be able to detect mutagens, to identify chemicals that can act as mutagens and therefore probably act as carcinogens. The best, the most reliable approach and the most direct approach is to assay point mutations directly. So there are lots of ways that we can do this indirectly by looking for mutant phenotypes and so on, but that's often very difficult. But the best approach for detecting mutagens is to develop an assay where we are directly inducing mutations and we can recognize when those mutations occur. So, you should have read about by now, I'm not going to take time during class. The Ames test is the most common test for detecting mutagens. It uses a microbial-based system, so it's quick, it's cheap, it's sensitive, it's quantitative. It fulfills all of the requirements for being able to run numerous samples through in a pretty quick period of time. Previous assays had included, for example, mice, which are another mammal. That's much more complicated because you can only grow so many mice. You can't get hundreds of thousands of them to make it quantitative. And it's very slow. The reproductive time on mice is a lot slower than the reproductive time on bacteria. So even though most mutagens that are identified in the Ames test are, in fact, followed up with mammalian tests, such as in mice, the initial screening is much faster and easier and cheaper with the bacterial system. And the bacterial system that Ames developed was Salmonella typhimurium. And your book has a long description. I think there's a screencast. This is just an outline of the process. It includes an adaptation to make bacteria respond more like you and I would respond, our metabolism would respond. And that's the incorporation of a liver extract. into the system. Your liver is a valuable organ. It detoxifies a lot of the things that we ingest that could potentially be harmful. The problem is that bacteria don't have livers. And so if we were simply assaying with a bacterial system, we're not necessarily assaying the way our metabolism would respond. And so that liver extract will incorporate a more accurate analysis of the effect of a potential mutagen by first having it exposed to liver enzymes that will modify or could modify that mutagen, that potential mutagen. And in some cases, it will make it less toxic, and after exposure to the liver extract, it will have no mutagenic effects. That's fine. That means that if we ingest it, our livers are going to do the same thing. In other cases, those liver enzymes could modify it in a way that, in fact, make it more mutagenic. And that's also useful to know because that's where our liver would not be doing the job it's supposed to do. Those enzymes would not be accomplishing what they needed to do, and we would want to limit the exposure to that particular chemical, even though enzymes are doing something, but it's making that mutagen toxic. Okay. So go through this, run through this. You have sample problems, I think. You have some examples to think through, make sure you understand the process here. There's also this website, I don't know if anybody tried to take a look at this, click on it, the virtual AIDS test. It's got a series of plates, and it has some conditions that you can modify, pull down menus to see a certain number of cells to start with, and then different compounds like caffeine and aflatoxin at different doses, and you can see in real time what the, in sped up, simulated AIDS test, what the impact would be of those particular chemicals on the bacterial growth. Notice that this is a direct test for inducing mutations, but it's not a forward mutation screen. It's not starting out with wild-type bacterial cells and asking, does this mutagen cause new mutations that produce new mutant phenotypes? That would be really, really hard to assay, because what would you be looking for? If you take a flask of wild-type bacteria and you pour this potential mutagen on it, what do you assay? Are you assaying for its inability to grow on some substance, its inability to make a particular item? You'd have to be doing dozens of individual assays on those cells. Instead, this is a screen for direct reverse mutations. So it's starting out with a mutant strain, something like an antibiotic resistance, and asking if we expose this mutant strain, which has a change in the DNA that is resulting in its resistance to some antibiotic, sensitivity to antibiotic. Let's start with the sensitivity, it's easier to screen. Then we expose that sensitive strain to a new mutagen, potential mutagen, and then plate it out on plates that have that antibiotic. The only cells that are going to grow are those that are resistant to that antibiotic, so have incorporated a reverse mutation. We started out with a mutant strain that is sensitive, and now we are looking for those that are resistant. And so the only cells that are going to grow on that plate are the ones that are the reverse mutants. This could be the same with an auxotroph, so some strain that requires methionine to grow, that's mutant. And we grow those strains, after exposure to the mutagen, we grow them on plates that do not contain methionine. The only cells that are going to grow are those that have reverse mutated to now be able to make their own methionine again and grow. So this reverse mutation aspect of it is a critical aspect of the approach because we don't have to think about all these different individual assays. We're simply looking for the ability of that mutagen, that potential mutagen, to cause mutations. And the type of original mutations, if it's a transition mutation, if that methionine minus strain is a transition mutation, or it's a frameshift mutation, something like that, then the assay can also tell us something about the form of action of that mutagen. If it can reverse transitions, we know what kind of a mutagenic action it has. If it can reverse frame shifts, we know what kind of a mutagenic action it has. Okay, so spend some time with this so that you understand it. How many of you have done this in microlab? You know, Okay, so it's easier when you have done it, and you have some sense of what's going on. So make sure you spend some time thinking through this process and recognizing how it's set up. Okay, so just spent a couple of days telling you all of the nasty things that can happen to your DNA. We knew about nasty things that could happen to the DNA just as a spontaneous error in DNA replication, just mistakes that happen. And now I've shown you all of these different mutagens that and agents that can cause those mistakes as well. The spontaneous error frequency during DNA replication is, okay, it's significant. It is less significant than it actually would be if there were not a series of pathways that are capable of recognizing mistakes and either fixing them or reversing them to reduce the persistence of those mistakes. So if we look at just a sequence here, if we start with the original error rate during DNA replication, it's something like 1 in 10 to the 5th nucleotides incorporated. But that rate is reduced by the ability of that polymerase complex to proofread during DNA replication. So that reduces it further. And on top of it, even after DNA replication is finished, there are what are referred to as post-replication mismatch repair processes that can catch some of those mistakes that didn't get caught during proofreading and restore the original nucleotide. And so we've gone from a 10 to the minus 5th to a 10 to the minus 10th error rate, that is mutation rate. So it's a lot lower than the error frequency that we start with. And that's because cells have evolved a number of different repair pathways that will fix those mistakes, recognize the mistakes, and fix them, whether they are mismatch mistakes or damage to the DNA. And only those that escape those repair processes persist and end up being among the mutations that actually get fixed in the population or get inherited. So when you think about this, we've got all these different repair processes, and we're able to fix some of them. Think about what would happen if we didn't have that. Right? If we had that 10 to the minus 5th error frequency during DNA replication alone, forget about the damage that we get from others. We'd have a lot more mutations. We'd have a lot higher frequency of cancer because mutations in cell cycle regulatory genes are the origins of most cancers. And as I said, most mistakes will lead to mutations in DNA functions. Any of those functions that regulate cell division are going to result in deregulating cell division, resulting in cancers. So this is a way of allowing some instability of the genome in order to get mutations that over evolutionary time might prove to be adaptive, but also to make sure that that level of mutation rate is manageable. It's manageable. So how do we measure then when we've got these various chemical mutagens? What are they doing to increase the mutation rate if we've got all these repair processes? Well, what they do is we saw with some of these, like increasing the probability of the tautomeric shift. It's increasing the likelihood of that kind of a mismatch happening. And when so many of them happen, the repair processes can't keep up. So as we'll see when we talk about some of these, the mutations will persist in spite of the repair processes because they're overwhelmed, they're saturated, and they can't keep up. If we expose ourselves to too high a frequency of mistake, the repair mechanisms just can't handle it. And that's how we have that differential between the so-called spontaneous rate and the induced mutation rate. That is, we expose ourselves to mutagens that are increasing that error rate at a higher frequency. Okay, so I'm going to talk about a number of these different repair pathways. Primarily, I'm going to focus on those that are referred to as error-free. They're referred to as error-free because they all incorporate the use of the complementary, undamaged strand of DNA as information as a basis for repairing the damaged DNA or the mismatched DNA. So there's some legitimate information that is being used in changing the damaged strand. There are the opposite of that. I don't think they're called error prone, but there are the opposite of that, which would just sort of randomly fix something. And you might think about this as the mechanism that allows a depurination to complete DNA replication. When we lost that G through depurination, there's no magical repair for that, except I'll tell you what one is that sometimes functions. But if that apurinic site persists, the DNA replication will go ahead and just incorporate an A opposite it. That's an error because it's not using any information in the nucleotide sequence to decide, I'm going to put an A in there. It's not being guided by complementary base pairing. It's just that there is no complementary base to follow. Okay. So we talk about the error-free approaches. The easiest way, the simplest way, most straightforward way you can imagine DNA repair happening is just a direct reversal. You've seen some of these. That's what happens during DNA polymerase proofreading during replication. If during the process of replication there is a recognition right at that time, at that spot, that a base has been misincorporated, that's easy. The proofreading mechanism will excise that base, maybe a couple around it, and replace it in the daughter strand according to the template strand nucleotide sequence. That's error-free. It's got complementary sequence, template sequence to use. So you're already familiar with that. Another one you're familiar with is photodimers, right? These photodimers that occur as a result of UV irradiation and the direct reversal. We have a repair process that simply breaks the dimer. It just breaks it apart, restoring the original two thymines that are adjacent to thymine bases. These are often referred to as pyrimidine dimers. You might have learned them as thymine dimers because that's a common dimer. But there's a whole collection of gene products, a protein complex that is involved in reversing these photodimers. At the heart of that is an enzyme called photolyase. This is the extent of what you need to recognize. I'm not interested in the details there, but this is an example of a direct reversal. The dimer makes a cross-linkage, and photolyase and its accomplices come and break it apart. Done. The alkyl transferases. The alkyl transferases are enzymes that can remove those alkyl groups that are put onto bases like guanine by alkylating agents, right? So this is one way to directly reverse the actions of that kind of a mutagen, those alkylating agents. So if we start with guanine and there's an alkyl group on it, there are specific alkyl transferases. There's a guanine alkyl transferase, and it recognizes that alkyl group on the guanine, and it cuts that bond, that covalent linkage that has attached the alkyl group to the guanine base, and it transfers it in a covalent linkage to itself, to a highly conserved cysteine in the enzyme amino acid sequence itself. So it's removing that alkyl group, and in doing that, it's restoring the normal guanine. So it's a direct reversal of that damage. The problem is that once that alkyl transferase transfers that alkyl group to its cysteine, it kills itself, right? It inactivates the enzyme, so the enzyme can no longer act. So now you can see how if you are exposed to a high dose of alkylating agents, at some point, very quickly, you're going to use up all of the alkyl transferases that are present in the cell, and the result will be a number of those alkyl guanines will persist and result in the kinds of changes that that mutagen is associated with. So we can't always have a direct reversal. So the next best thing is to be able to identify and then remove and replace the damage or the mismatch. So I want to talk about a number of systems that are associated, or processes, repair pathways that are associated with this removal and replacement. So it's not a direct reversal, but it's rather cutting out the region that has the damage or the mismatch and using the complementary strand as a template to replace it with the normal sequence. There are two in particular I want to talk about: base excision repair and nucleotide excision repair. So these are lumped under the category of excision repair pathways. And the base versus nucleotide is just simply identifying, making the distinction between what is excised. In the base excision repair, only the base is removed, not the whole nucleotide. And obviously with the nucleotide excision repair, the whole nucleotide is. So here are two examples of damage that can be repaired by the base excision repair. Remember we talked about uracil ending up in DNA because of what? How might you encounter uracil in DNA? It's not supposed to be there. Hold on. When you relay this back, I'm throwing it to you. Whoops, and I'm not really good at that. Gotta get rid of things in my hands.
Deamination of cytosine.
Deamination of cytosine. Remember, deamination of cytosine produces uracil. Uracil is recognized by a uracil glycosylase, right? Like the glycosylase we just talked about, like the alkyl glycosylase. So the uracil glycosylase will remove the uracil, and then it will replace it. It will synthesize a new strand so that the uracil is no longer there, the cytosine will be there. So the recognition part of this is the uracil, the excision, the base excision is with this glycosylase, and then that's followed by an endonuclease called an AP endonuclease, which will synthesize the new corrected, original, unmutated strand. So these excision repair processes, the base excision is two parts. It includes a glycosylase to break that glycosidic linkage, the one that connects the base to the sugar, and followed by an endonuclease. An endonuclease which is going to remove sequences around that excision and repair them. All right, so here's a diagrammatic representation. The damaged base is, let's say that this is the damaged base that's blue here, and the glycosylase will recognize that this is an incorrect base, and it will cut the glycosidic linkage. Notice that the DNA backbone, the sugar phosphate backbone, is not broken. The phosphodiester bonds are not broken at this point, but the release, that breakage of the glycosidic linkage and the release of the base recruits to the site an endonuclease, referred to as an AP endonuclease. It's AP because this is going to result in what we call an A-basic site, either an A-purinic site or an A-pyrimidinic site, hence AP. This is an AP site that has lost the base, and that might be a purine or a pyrimidine. Which one is it in this case? A purine, because it's the bigger one, right? Or however you use your mnemonic. So when this glycosylase releases that base, it's gone. It's left an apurinic site in this case, and that apurinic site is recruiting, is target for an AP endonuclease. At that site, that AP endonuclease makes a cut. It's a nick. Whenever we talk about cutting the DNA on only one strand, the term we use is a nick. So it's nicking this DNA only on this strand. It's an endonuclease because it's cutting, it's a nuclease, but it's cutting within the DNA strand, not on the end. So it's not an exonuclease, which would be digesting from a free end. It's an endonuclease, it's cutting through the middle. And then this endonuclease cut here exposes two free ends that are followed, are recognized by an excision exonuclease. Here's a cut end, and it's going to digest on either side of where that AP site was, making a region here that has no nucleotides at all. So it's got only that single strand. That single strand is then going to serve as the template, for polymerase. That polymerase is going to synthesize the new daughter strand here in the red nucleotides based on the complementary sequence on that complementary strand. So this is why this is error-free. It's got information. It's actually building that repaired strand based on some legitimate information. This whole thing referred to, that includes the excision and the repair, is referred to as an excision repair cassette, basically, through excision repair pathway. In this case, it's a base excision repair, but it can also be a nucleotide excision repair. So in that case, this could be relatively subtle problems like a mismatch that resulted after tautomeric shift. Remember, we sometimes will have that hybrid DNA double helix where it's a mismatch after the base has gone back to its normal isomer, and if sometimes that's recognized as a little bulge or hydrogen bonding isn't quite right, and it could be recognized by the base excision repair. The glycosylase will show up here and maybe release that. Sometimes it is a matter of some more subtle damage to the base. Maybe it's subtle damage that is like the alkylation, but not maybe as dramatic as that. Maybe glycosylases that are specific for uracil could be, you have a counterpart here if you think about it in other nucleotides, other bases. So this is just releasing that base. More severe damage, so that's exonuclease, polymerase, and ligase, is that excision repair. This is just like DNA replication. Polymerase is a special polymerase, but it polymerizes based on complementarity, and then that last phosphodiester bond has to be accomplished by ligase because polymerase can't do that. Okay. I've said all these things. Uracil glycosylase removes the U formed by C deamination, and these AP endonucleases can repair apurinic sites, like the one that we used as the example to show the origin of transversion. So if that did not get recognized here, it would persist as an apurinic site, and there would be an A put in opposite that site during the completion of replication. But if it's recognized early, it might be recognized by this excision repair, base excision repair. Okay. The other flip side of this is a nucleotide excision repair. And so this might be more significant damage to the DNA. And there are two ways in which this damage is usually identified or recognized because of the consequence to that damage during either DNA replication or transcription. So again, this is an excision repair process, only instead of eliminating just a base followed by excision, it's eliminating a nucleotide. Okay. So some of the kinds of damage that are pretty significant, aflatoxin. You've probably heard of aflatoxin, pretty strong mutagen in a number of mushrooms, fungi, really, really deadly. And this is aflatoxin that sticks itself onto the sugar in the DNA backbone, makes a bulky region in that DNA, which is significant damage, and it needs to be replaced. So nucleotide excision repair. Keep in mind what you just saw for the base excision repair and do some comparing and contrasting. All right, we have to recognize the damaged DNA. All right, not so hard when you've got something like that stuck on it. And now there's a big complex. It's not simply an enzyme like glycosylase. It's a big, huge complex. I don't care that you know any of the components of this complex, but to recognize that this is a major molecular collaboration with a large number of proteins. And that damage then recruits the assembly of that multiple protein complex to that site of the damage. And then it's going to cut out the damage plus some order, some freedom on either side to make sure that the whole thing is cut out. It's going to cut out a whole segment of that nucleotide strand. So it's not releasing a single base or any bases, it's cutting out the nucleotides. And then again, because it's error-free, it's using the complementary strand as a template. So two ways that this can be recognized. The two processes that would be most disrupted by significant DNA damage are one, DNA replication. If you've got a big clunky old addition or damage to DNA site, DNA polymerase complex isn't going to be able to synthesize through that site. It's going to get stuck. So stalled replication complexes are often a trigger for assembly of the nucleotide excision repair complex. Another way that it's recognized is during transcription. Right? You know enough about nucleotide synthesis now to know that those two processes are really, really similar. Both require polymerase complex, whether DNA or RNA, and both have to separate the two strands of the nucleotides in order to synthesize the new strand, whether it's a DNA complement or the RNA transcript. And so that kind of stalled transcription complex can also trigger the nucleotide excision repair. Which one of those is identified? They each use a slightly different recognition complex, but the end result is the same. So they're separated here to show that it's either recognized as a stalled replication complex or a stalled transcription complex. And that both of above those events will trigger the assembly of this nucleotide excision complex. Notice the participation of an old friend, TF2H. We've seen TF2H now participate in a variety of different molecular events, and it makes sense here that if we're going to cut away a significant part of that damaged single strand, we're going to need to separate those two strands. And so the TF2H is part of what will recruit this assembly of the multi-protein nucleotide excision repair complex. So it comes to this site, here's the damage, here it's stalled. It might be a binding dimer. It might be something like that. It escapes the DNA and prohibits that complex, the replication or transcription complex, to proceed. So it assembles on this site, and it spans quite a bit around that original site of damage. And then the result is to remove the damaged DNA. So cutting on either side within this complex, this is again an endonuclease. It's making a nick on either side. So it's making two nicks that flank the damage. When that happens, basically the only thing that's holding that sequence onto that DNA molecule are those hydrogen bonds, and it's not easy to separate them and pull them away. This is maybe 30 bases total, somewhere along there, that's the order of magnitude. It's not hard in the context of a whole DNA molecule to pull those 30 bases in that single strand nucleotide as part of that complex that's assembled there and is removing that damage, to just take it and throw it away. That then recruits this bypass polymerase. It's a special polymerase that is optimized for short distances. So this is a polymerase, a DNA polymerase, that would not function during normal replication because it wouldn't stay on the DNA template long enough to do that. It's really maximized, optimized for very short stretches. We have that same synthesis here. It gets to the other end, and just like any other polymerase, it can't make that last phosphodiester bond, and so ligase makes that last phosphodiester bond. And the result is having excised that damage and replaced it with the normal sequence, using the complementary strand information to tell us what that normal sequence should be. All right. So again, this repair synthesis, you'll sometimes see reference to repair synthesis all by itself. This collection of components, the bypass polymerase with its clamp that keeps it on the DNA for that short stretch, and ligase, is often referred to as a cassette that can follow any other kind of repair where excision is happening and it needs to follow up on it. You should see some similarities from one of these processes to the next, because we're basically not recreating a whole new repair mechanism for each one, but we're using components of it. There are like cassettes, mobile cassettes that can function here or here or here, and yet there are also individual specific components. Okay. All right. So most of the time we're looking at this, and for example, this nucleotide excision repair, if it's stalled during replication, obviously replication is still ongoing because it's stuck. And we've seen some of the other cases where when a mismatch is caught, like proofreading, or that initial recognition with a glycosylase base excision repair is caught during the process of DNA replication and can be fixed. But sometimes that damage persists, or the mismatch persists, beyond the completion of DNA replication, at least for that segment of the chromosome of the DNA molecule. And so there's another category of error-free repair mechanisms that's referred to as post-replication. Namely, it doesn't have to be caught in the act of replicating, but it's still possible to find that damage or that mismatch and fix it. One category of subcategory of this post-replication repair is mismatch repair. And that's the one I want to talk about. Okay? Because this is the kind of repair that, in fact, catches most of the mismatches, the missed incorporations during DNA synthesis that proofreading doesn't catch, and it's actually modeled on the proofreading, except that it can happen after the polymerase complex is no longer associated with that DNA after replication is finished. 3 steps. Should be kind of intuitive to you by now. You have to obviously recognize the mismatch base pair. If I've got a CA base pair, there's something wrong there. I've got to be able to recognize that that's a mismatch. Then you have to determine in that mismatch, if you're now after DNA replication, how do I know which one's wrong? if I've got a CA bulge mismatch, this hydrogen bonding a little bit off, and so it's recognized as not quite right, there's a mismatch, how do I know which one is correct? Is it the C or is it the A? Should it be CG or should it be A? That's the key. So we have to then determine which of the bases is incorrect. And then once we have determined that, you get rid of it and carry out repair synthesis. You get rid of the base and carry out repair synthesis. So that's the most important step in this process, is when you've got a mismatch out of the context of the actively replicated DNA. The repair, it might be still the molecules replicating, but that fork has moved on, and now all you're looking at is a double-stranded DNA. Maybe the rest of the molecule is still replicating. You've got that stretch of DNA that you just looked at it. How are you supposed to know which one was template and which one was daughter strand? So that's the key, because you might repair it in the wrong direction. So finding out which base, identifying, recognizing the base that's incorrect is key. This is best understood in bacteria, where it's been studied the best, the most, and recognizing that the errors that occur are going to lead to mismatches in the newly synthesized daughter strand. So if a mistake happens during DNA replication, nothing's happening to the template strand. That's just there serving as the model, the complementary strand model. That if a mistake has happened and you end up with a mismatch, the wrong base is going to be the one in the daughter strand. So the whole key here is when you're looking at that double-stranded region, which one's template, which one's newly synthesized daughter. And that's the basis for the mismatch repair in bacteria. Bacteria can recognize the old and new strands based on modification of those strands that is methylation. So different nucleotides can be methylated. In bacteria, the critical nucleotide is an A. And the sequence in which that A is methylated is GATC. So various GATCs occur in the genome of bacteria, and when they do, the A is found to be methylated. So now when we look at this, here's the fork, it's going through and sequence, the fork is moving this way, right? And so we've got the sequence that's already replicated. Here, the daughter and the template strands are nicely marked for you, blue and yellow, mustard color. But not true in vivo. How do we know? If I'm looking over here, how do I know which one is the daughter and which one is the template? Well, there are a variety of these GATC sequences along the genome. And notice, this is the double-stranded sequence. It's the same in both directions, right? GATC 5', the other strand is GATC 5' to 3', right? So we've got this palindrome, and in the non-replicating genome, this A would be methylated, and this A would be methylated. But for a brief period of time, and these methylations are accomplished by an enzyme called adenine methylase, and that goes to these adenines specifically. Right at this point, this is the most recent thing synthesized at this fork, and for that brief period of time, the A's that are incorporated are just normal ANTPs, GATPs. They're not the methyl ATPs, they're ATPs. The methylation is accomplished after the fact by this enzyme. So for some short period of time after that synthesis has taken place, these A's on the daughter strand are not yet methylated. This molecule here, as it's been synthesized, is heavy methylated. It's only got one of the strands methylated. And so this system in bacteria will recognize that there is a hemimethylated double-strand region here. That means that identifies this strand that's unmethylated as the daughter strand. This strand is the daughter strand. I've got to replace this T in the mismatch. The real base should be a GC because this was the new strand and that's where all the errors have happened. I've now got to replace that T with a C to be a normal base pair. Okay. It's not clear in eukaryotes. It's not completely understood how this mismatch repair functions in eukaryotes to identify the new daughter strand. There is methylation in eukaryotes. It's primarily methylation of cytosine nucleotides, but there's some eukaryotes that don't have any methylation of Cs at all. And in humans, there's a very skewed bias towards where the Cs are methylated. So it's not completely clear, it's not likely that cytosine methylation is, if it's acting at all towards helping in this identification, it certainly can't be acting as the sole, maybe not even the primary mechanism. So then we follow with excision and repair, so same thing I mentioned before. So the question is, how do we identify the old and new strands in eukaryotes? So picture a situation like this. So we've got this fork, and the fork is moving, the bubble is expanding, and in this bubble region, there's already synthesized double strands. So during DNA replication, what kind of feature might you look for in eukaryotic replication that could be used to help you distinguish the old versus the new strand, the template versus the daughter strand? Any ideas? Remember reading it? What do you always see in some regions of the genome during DNA replication on the new strand and not on the old strand? I'm going to go back and review. Right? If I've got this bubble and the forks are moving like this, that on every DNA replication, whether it's using the bottom strand as a template or the top strand, the daughter strand, the lagging regions, the lagging strand regions of the daughter strand are going to have those Okazaki fragments. And remember that those Okazaki fragments do not get sealed with ligase immediately. There is a lag, just like there's a lag with the adenine methylase after the synthesis has gone through that region. And so for some brief period of time, this region here has got a number of free 3' hydroxyl ends in that daughter strand that may be recognized as saying, okay, I know that this strand now is the daughter strand. So if I've got a mismatch over here, this is the strand I need to replace nucleotide excision repair, in case excision repair. So that's one of the current models of how eukaryotes recognize that difference between old and new. So I want to leave you with this last bit, that we've seen all these different DNA repair mechanisms. They're really, really important. If we didn't have them, we'd have a much higher rate of mutation. But all those repair mechanisms are run by gene products, right? Proteins that assemble to do all of these. And so mutations in those DNA repair process genes result in disorders. And those are often identified as cancer syndromes, aging syndromes, where stability of the DNA leads to breaks and mutations, premature aging because the cells start to have DNA fragmentation and start to die. So a number of these different disorders are consistent. I've got this table here to show you which repair process is mutated, has gene mutations associated with it. You've probably heard of some of these before, xeroderma pigmentosa, and so on. So next time we're going to start talking about inheritance patterns, what we call xenogenetic genetics. But before we get there, we got to do meiosis first.